Or was I? Sorry, na brown, na utod. Okay, so next. So for RNA viruses, we have three types: positive stranded, negative stranded, or the RNA retroviruses. For positive, like the messenger RNA, when it enters the host, it can immediately translated by the host ribosome into protein. So if the virus, if the RNA is already positive, it's already like the messenger RNA, then it can produce or it can reproduce or multiply its copies into new viruses. So if it's negative, it must be transcribed first into a positive strand. They should carry their capsid and enzyme called RNA-dependent RNA polymerase, which carries the transcription of the negative strand into a positive strand. Ano is, if positive siya, di ba? The same as with your mRNA, diretso na multiply or reproduce some viral copies. If it's negative, it should have an RNA-dependent RNA polymerase to transmute the positive into negative, of the negative to the positive, sorry. Once positive na siya, again, like the messenger RNA, it enters the host and immediately be translated the host ribosomes into proteins, leading to new viral copies. Okay? However, the RNA retrovirus, the RNA of the retrovirus is transcribed in a reversed fashion into DNA. So RNA should be converted first into DNA. So the enzymes that is responsible for doing that is the reverse transcriptase. If it's already a DNA, your DNA or codon DNA, your DNA has already the positive strand and the negative strand. Only the positive strand is being used to transcribe new viruses. Okay? So for the DNA viruses, they cannot be translated directly into proteins and thus be translated into mRNA. However, they already have both the negative strand and the positive strand. So only the positive strand is used as a template for transcription into mRNA. So this is the viral picture or the viral morphology. The protein shell or coat that encloses the nucleic acid and the genome. So it have two types, the icosahedral and the helical. So guys, take note. This is the genomic or genetic material. Okay? This is the one that surrounds it is your nucleocapsid. This is the envelope. This is your spike. Okay? Here, this is the genetic material or the nucleic acid or the genome. This is your nucleocapsid. How about this one? Susunod. These are called your capsomeres. Ang isa-isa niyo. These are your capsomeres. This is also your spike. Okay? Same thing. So capsomeres. Capsomeres are microscopic units of the surface of an icosahedral virus particle, and it represents a cluster of polypeptides, but the morphologic units do not necessarily correspond to the chemically defined structure units. So your structural units are also called as protomere, a basic protein building blocks of the coat, and a collection of more than one non-identical protein subunits. So you have your helical symmetry capsids. The protein capsomeres are bound to RNA and called into a helical nucleoprotein capsid. Most of them assume the spherical shape except for your rhabdoviruses, which have bullet-shaped capsids. Your rhabdoviruses are your rabies virus. So the envelope, the lipid bilayer membrane structure that covers capsid that contains the nucleic acid. Viruses acquire this membrane by budding through the host cell and the nuclear membrane entering up a piece of the membrane as they leave. So primarily of cellular in origin with virally coded proteins on the surface. So guys, tandaan, ang envelope halina siya sa host cell. However, may ara lang gid viral coded proteins sa iya nga surface. So envelope are fragile in such that anything can disrupt their envelope that inactivates the virus. So it contains enough enzyme to start replication without relying on the host cell. Viruses that do not have the membranes are called naked or non-enveloped. So when we say naked viruses, these are the virus that contains only the virion. When we say virion, it's just the nucleocapsid and the genome. However, naked viruses, as mentioned earlier, they are relatively stable to temperature, pH, and chemicals. So the takeaway notes, this is the classification of viruses for the nucleic acid, either DNA or RNA, double or single-stranded, segmented or non-segmented, positive or negative stranded, and the complexity of the genome. If it's capsid, either icosahedral, helical, bullet-shaped, spherical, or even complex shape. The number of capsomeres, the size of the capsid or the box is 250 by 350 nm, while the polio is 25 nanometers. We can also have the type of host it infects and the type of disease it produced, and of course the target cells. As mentioned earlier, target cells or host cell is specific for viruses. Okay? Cell specific po siya. And of course, immunologic properties. We can also classify viruses according to its envelope, either enveloped or naked, and the size of the helical capsid viruses and the number of capsomeres in your icosahedral capsids. So these are your RNA viruses. RNA and DNA viruses. Sa babaw is the DNA, the poxviridae, asparviridae, herpesviridae, adenoviridae, papovaviridae, parvoviridae, circoviridae, hepadnaviridae, and retroviridae. While on the lower part are the RNA viruses. Next, we have the viral replication. This is a representation of all the change with time in the amount of infectious virus in a single cell that has been infected by a single virus particle. So in practice, this is determined by following events in a large population of infected cells in which the infection is proceeding as nearly synchronously as can be achieved by manipulating the experimental conditions. Time scale and progeny virus vary greatly among virus family, but the basic features of the infectious cycle are very much similar. So we also have the term eclipse period, which is following the initial attachment of a virus to a host cell. It is the ability of the virus to infect other cells disappear. So meaning to say, when we say eclipse period, ang imo virus nakasulod na sa isa ka cell. Since nakasulod na siya or naka-attach or nakapilit sa isa ka cell, hindi siya pwede mang-infect other cell. Meaning to say, one virus, one cell. So this is the time elapsed from initial entry and disassembly of the parental virus to the assembly of the first progeny virion. At this period, active synthesis of virus components is occurring. For most human viruses, this falls within a range of 1 to 20 hours. Okay? If we have the eclipse, we also have the exponential growth. So when we say exponential growth, this is the number of progeny virus produced within the infected cell increases exponentially for a period of time and then reaches a plateau, after which no additional increase in virus yield occurs. So meaning to say, when we say exponential growth, ang pinakadamo or ang peak, ang pinakapunta na kung ano kadamo ang virus na ma-produce niya. That's your exponential growth. So the maximum yield per cell is a characteristics for each virus cell system. That reflects the balance between the rate at which virus components continue to be synthesized and assembled into virions and the rate at which the cell loses the synthetic capacity and structural integrity needed to produce new virus particles. So this may be formed 8 to 72 hours or longer that yields 100 to 10,000 virions per cell. So last slides. So these are the steps in the replication cycles of viruses. First, there is an absorption or attachment. A virus attaches to the surface of the cell by means of specialized structures on its surface and specific receptors on the surface of the cell. Then, after adsorption or attachment, there is penetration. This is the taking over of the host cell's machinery, wherein the virus enters the cell by endocytosis, meaning to say, ginkaon or gininom siya sang cell. So the fusion of viral membrane and cell membrane or lysis of the membrane, or either ang imo man mismo nga virus ang nagpursige man nga ma-penetrate niya ang imo nga cell. In bacteriophage, or simply saying as phage, these are the viruses that infects the bacteria. Penetration happens when the phage injects its DNA into the bacterial cell. After that, there is already an uncoating. Once inside the cell, the virus breaks down into its component parts, wherein viral nucleic acid escapes from the capsid. Take note, in bacteriophage, there is no uncoating that happens. Why? Kaya ang bacteriophage wala gasulod sa sulod ng cell. Gina-inject yan ang genetic material niya, while most viruses goes inside the cell. Next, for biosynthesis, the virus directs the host cell to make viral components, and the metabolism of the cell may be shut off completely like that of your poliovirus, or it may continue, however, on a restricted scale, such as your influenza virus and papovavirus. The cell shumps out viral components until there are enough for viral assembly to start. So during this step, the host cell enzymes, nucleotides, amino acids, and ribosomes are used to make viral DNA and viral proteins. After that, we will have the assembly that involves fitting of the viral pieces together to form a complete virion. So the components are assembled within the cell and moved to the cell surface. So they can also be released in two ways. First, by lysis of the cell, like the poliovirus, or by budding through the cell membrane to acquire the envelope, like your influenza and para-influenza virus. Okay? So that would be for now. For the introduction to virology, I will continue my discussion next meeting and upload pre-recorded videos. Thank you.